Good morning, children. Hope all are fine. I welcome you all to our another class of botany. In our first chapter in botany, that is the living world, till now we have studied about attributes of living world. Next, we studied in detail about virus. Today, we are going to study about classification of living world. One thing I want to say here is before studying classification, we have already studied virus. Do you know why? Yes, you know the reason very well because virus exhibit both living and non-living features. So it is kept in the classification of living, it is kept in between the living classification of living and non-living things. So classification of living world. From the previous chapter, we know that the planet Earth is endowed with living and non-living things. So we have different types of living and non-living things on this planet. In our daily life, we see several things in and around us. Imagine you are on a trip to hill station. You are enjoying the beauty of mountains, dazzling colors of the flowers, and melodious sound of the birds. You may be capturing most of the things you come across in the form of photography. Now, from this experience, can you mention the objects you have come across? Can you record your observations and tabulate them? How will you organize the things? Will you place mountains and flowers together, or tall trees and trailing herbs in one category, or place it in different category? If you place in different category, what made you to place them in different category? So classification is essential and could be done only by understanding and comparing the things based on some characters. Similarly, all of us have visited a textile shop. In a textile shop, we can see the cloths are arranged in a particular order, like depending on the texture of the cloth. Next, the range of price, age group, gender, etc. So what is the use of this classifying these cloths in different category? This help us to make our selection easier. Here also, for studying the organisms of living world, classification is essential. Many attempts have been made in the past to classify the organisms on earth. Theophrastus, who is said to be the father of botany, used the morphological characters. Morphological characters means external characters to classify the plants. So based on the external characteristics of the plants, like height of the plant, okay, the plants were classified into three. They are trees, shrubs and herbs. Aristotle, he classified the animals into two groups. Okay, as we studied Theophrastus, he classified plants into three groups. Similarly, Aristotle classified the animals into two groups based on the color of the blood. So it was classified, the two groups were Enaima and Anaima. Enaima, that include all the animals which had red blood, and Anaima, it included the animals without the red blood. Next, Carl Linnaeus, he classified the living world into two groups, namely plants and animals. So here the Carl Linnaeus, his classification is said to be a two-kingdom classification. So he classified all the living organisms of the world into two groups, that is the plant kingdom and the animal kingdom, based only on the morphological characters. His classification faced major setbacks. Okay, the Carl Linnaeus classification, there were many drawbacks of this classification. Two drawbacks is being mentioned here. So first drawback here, it is the prokaryotes and eukaryotes were grouped together. Now what are these prokaryotes? Pro means it's a initial stage, or which is in a state of development, or which is not well developed, and karyotes refers to nucleus. So prokaryotes means those organisms which do not have a well developed nucleus. Next, eukaryotes. The word EUU means true. Again, eukaryotes refers to nucleus. So here, though eukaryotes means those organisms which have a well-developed nucleus. So the prokaryotes and eukaryotes were kept together in one group and in called Linnaeus classification. So this was one drawback for this classification. Similarly, fungi, a heterotrophic organism. All of you have seen the fungi. A common example you can take mushroom. The mushroom we can say it is non-green in color. Okay, pale white in color. Okay, so this as we know, since they lack chlorophyll, they cannot prepare their own food. So they depend on other organisms for their food. So this mode of nutrition we call it as heterotrophic. So fungi, they are heterotrophic organism, and this heterotrophic organism was also placed along with the photosynthetic plants. So here Carl Linnaeus classification, some feedbacks were there, some drawbacks were there. So among that drawbacks, two drawbacks is, first one we studied prokaryotes and eukaryotes were kept together in one group. Next drawback we studied fungi, which is having a heterotrophic mode of nutrition. It was kept with plants which have an autotrophic mode of nutrition. So in the course of development, in the course of time of classification, various fields of biology was also taken into consideration for classification. So here we can say the earlier form of classification was based on morphological characters, external characters. So later form of classification, here the information from other field of science was also taken into consideration, like cytology, anatomy, embryology, molecular biology, phylogeny, etc. Cytology means study about the cells. Anatomy, internal structure. Embryology, study development of the embryo. Molecular biology, that is the molecules which is present inside the cell, or the biomolecules which are found inside a organism. Next, phylogeny, the evolutionary history. These were also taken into consideration for classification of the organisms on earth. Next, we are going to study about the need for classification. Why these organisms has to be classified? So first need for classification is to relate things based on common characteristic features. So first necessity is to categorize, to form an organism, to relate the things based on common characteristic features. We are classifying the organisms based on the common features. So those organisms which have similar features, they are kept together. Those organisms which have dissimilar features, they are kept separately. Next, to define organism based on salient features. Each organism has their unique or particular characteristics. So based on that, these organisms were separated or they were grouped. Now helps in knowing the relationship among different groups of organisms. From classification, we can develop a relationship between the different organisms. Next, it helps to understand the evolutionary relationship between the organism. So it also helps us to know how these organisms has evolved, from where this evolution of the organism has taken place. That can also be studied with the help of classification. Now, classification of living world. A comparison of classification proposed for classification of living organism is given here. So here you can see different scientists, they have given different kingdom classification. So first one we studied Carl Linnaeus, he gave the two kingdom classification in the year 1735. So the two kingdom were kingdom Plantae and kingdom Animalia. Later in the year 1866, Ernest Haeckel, he classified three kingdom classification. One more kingdom was added up here. That kingdom was Protista. This Protista included both unicellular eukaryotes as well as the prokaryotes. Next, after few years, four kingdom classification was given by Copeland in the year 1956. So what he did, he separated the eukaryotes and the unicellular eukaryotes and prokaryotes, and he gave a four kingdom classification. So that was kingdom Monera, kingdom Protista, kingdom Plantae and kingdom Animalia. Kingdom Monera, it included unicellular prokaryotes. Kingdom Protista, it included unicellular eukaryotes. Kingdom Plantae and kingdom Animalia, it included the multicellular organism. Later, the five kingdom classification was given by R.H. Whittaker in the year 1969, and here he separated the he gave a separate kingdom position for fungus. So he classified five kingdom classification. So first one, kingdom Monera, Protista, fungi was separated from Plantae. So separate kingdom status was given for fungi. Next, Plantae and Animalia. So here in our syllabus, we are going to study five kingdom classification in detail. The five kingdom classification was given by R.H. Whittaker, an American taxonomist. Okay, he proposed the five kingdom classification in the year 1969. So this five kingdom, it included kingdom Monera, kingdom Protista, kingdom fungi, Plantae and Animalia. Here we will study the comparison of these five kingdoms. So here you can find, so there are certain criteria for the classification of organism. So first criteria is cell type. So here Monera included the prokaryotes, Protista, fungi, Plantae and Animalia, they all included the eukaryotes. Next, level of organization, how these cells are present. So here Monera, mostly they were unicellular, rarely they were multicellular. So most of the organisms in the kingdom Monera, they were unicellular, very few multicellular. In Protista, it included the unicellular eukaryotes. Okay, fungi, it included both unicellular as well as the multicellular non-green organisms. Kingdom Plantae, it included those organisms which have the tissues or the organs. Okay, next Animalia, it included those organisms which have tissue, organ systems were present there. Next criteria of the classification is cell wall. So here in the kingdom Monera, they had the organisms in which cell wall was present, and the cell wall, it was made up of peptidoglycan and mucopeptides. In Protista, organisms, they have the cell wall, and the cell wall is made up of cellulose. Okay, in some only the cell wall is present, in others it is absent. Here another feature we can say, in the Protista, unicellular eukaryotic protozoans. Okay, protozoans were also there, and phytoplanktons were also there. Small plants and small animals, microscopic plants and animals were included under the Protista. So here, next, cell wall, kingdom fungi. In this, cell wall is present, and the cell wall is made up of either chitin or cellulose. Next, kingdom Plantae, cell wall is made up of cellulose. Animalia, cell wall is absent. So those organisms which lack the cell wall, they were brought under the kingdom Animalia. Next criteria for classification is nutrition. So whether they are autotrophs or they are heterotrophs. So in the kingdom Monera, they were autotrophic organism. Autotrophic also, they have two modes are there, phototrophic and chemotrophic. Phototrophic means those organisms which use the sunlight for preparation of food. Chemotrophic means those organisms which use the chemical energy for the preparation of food. So either in chemotrophic way or in the phototrophic way, the organism prepare their own food. So such type of organisms were brought under the Monera. It also included some heterotrophic organisms. Next, Protista. Here the organisms, they were autotrophic, photosynthetic and some were heterotrophic. As we studied, they included the protozoans as well as the phytoplanktons. Next, kingdom fungi. Kingdom fungi, here the mode of nutrition is heterotrophic. They cannot prepare their own food. Next, Plantae, the mode of nutrition is autotrophic. They prepare their own food by the process of photosynthesis. Next, the last kingdom, Animalia, the nutrition is heterotrophic. Animals, they cannot prepare their own food, so they have the heterotrophic mode of nutrition. Next, their motility. Okay, motility means they can move, the ability to move from place to place. So Monera, they were motile or non-motile. Protista, again they were motile or non-motile. Fungi, they were non-motile. Plantae, again mostly non-motile. Animalia, mostly motile. So some examples of each kingdom. So here first one, kingdom Monera, they included Archaebacteria, Eubacteria, Cyanobacteria, Actinomycetes and Mycoplasma. Here Protista, the organisms which are included under Protista, they were chrysophytes, dinoflagellates, euglenoids, slime molds, amoeba, plasmodium, trypanosoma and paramecium. Fungi, examples yeast, mushrooms, molds. Plantae, it include algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Animalia, all the sponges, invertebrates and vertebrates, they were included under the Animalia. So this was the 5 kingdom classification as given by Whitaker. Now in this 5 kingdom classification, there were the criteria for the classification as we are going to study, as we have already studied. Again we can see here the criteria adopted for the classification, they included cell structure. So cell structure, whether they are prokaryotes or eukaryotes. Thallus organization. Thallus organization means cellular organization, or we can say the plant body, whether it is unicellular or multicellular. Next, mode of nutrition, whether they are autotrophs or they are heterotrophs. Reproduction. Reproduction, either they are reproducing asexually or they are reproducing sexually. Next, phylogenetic relationship. Their evolutionary history was also taken into classification for giving this 5 kingdom classification. A comparative account of the salient features we have already studied here. In this classification, the 5 kingdom classification also, there were some merits and demerits that we are going to study next. So merits of this classification. The classification is based on the complexity of the cell structure and organization of the thallus, as we have studied. The classification is based on the nature of the cell, whether they are prokaryotes or eukaryotes. Prokaryotes and eukaryotes, they were kept separately in the kingdom. Next, their cell organization. Okay, the complexity of the cell, whether they are unicellular or they are multicellular. Based on that also, these organisms were classified. So here, first merit of this classification was, classification was based on cell structure and organization of thallus. Next, it is based on the mode of nutrition. So here also the organisms, the autotrophs and the heterotrophs were kept separately. Next, separation of fungi from plants. As we studied, fungi, they show an heterotrophic mode of nutrition and plants, they are autotrophs. So fungi was separated from the plants and given a separate kingdom status. Next, it shows the phylogeny of the organisms. So phylogeny, this classification is explaining the evolutionary history also. As we can see, unicellular simple organisms are kept in the beginning of the classification, and the most complex multicellular organism, the plants and the animals, they are kept in the end of the classification. So this classification was also explaining the phylogeny of the organisms. Now we are going to discuss about the demerits of this classification. So the first demerit of this classification, the kingdom Monera and Protista accommodated both autotrophic and heterotrophic organisms. As we already studied, Monera and Protista, both autotrophs and heterotrophs were kept together. Cell wall lacking and cell wall bearing organism, thus making these two groups more heterogeneous. So as we have studied in Protista, we have studied phytoplanktons are the phytoplanktons means small aquatic or plants, okay, and protozoans means small aquatic animals. So as we know, plants they have the cell wall. So phytoplanktons they have the cell wall, and protozoans they lack the cell wall. So those organisms having both the presence and absence of the cell wall, they were brought together under the kingdom Protista. So here this may, as a result of this, Monera and Protista they had a wide boundary. So it made more heterogeneous to this classification. That was one drawback for this classification. Another drawback for this classification was virus was not included in the system. As we know, virus they exhibit both the living and the non-living features. So this virus was given a separate position. It cannot be classified under the living and the non-living. So it is kept in between the living and the non-living organisms in the classification. So here we have studied in detail about the 5 kingdom classification. The 5 kingdom classification as given by R.H. Whitaker. So classification was still going on. Classification process, the scientists were still working on this. So further they have classified this organism. They have tried to classify this organisms of the living world. So Carl Woese and co-workers in the year 1990, they introduced 3 domains of life. So domains they are at a higher level than the kingdom. So what they said, all the living organisms they can be classified into 3 domains. So what are the 3 domains? They are the bacteria, archaea and eukarya. So they said the organisms cannot be kept classified into kingdom. So we can classify them into domains. So they classified all the living organisms in 3 domains. So bacteria, archaea and eukarya, based on the difference of recombinant DNA nucleotide sequence, lipid structure of the cell membrane. So here, so based on the nucleotide sequence as well as the cell membranes, nature of the cell membrane, the lipid structure of the cell membrane. After this, a revised 6 kingdom classification was given for the living world. This was proposed by Thomas Cavalier Smith in the year 1998. So, as we have studied 5 kingdom classification given by R.H. Whitaker. So, from that 5 kingdom classification, Thomas Cavalier, he has classified this 5 kingdom further into 6 kingdom. So, what he did, the kingdom Monera was divided into Archaebacteria and Eubacteria. So along with this, what we studied 5 kingdom classification, Monera, Protista, fungi, plantae, animalia. So here Thomas Cavalier, what he did, he further divided this Monera into 2 kingdoms. They were Archaebacteria and Eubacteria. Recently, Ruggiero et al. 2015 published a 7 kingdom classification, which is a practical extension of Thomas Cavalier's 6 kingdom scheme. So here from the 5 kingdom classification we got the 6 kingdom classification. The 6 kingdom classification further it was classified into 7 kingdom. So according to this classification there are 2 super kingdoms. So they said much higher. There are 2 super kingdoms. They are prokaryota and eukaryota. These prokaryotes they include 2 kingdoms, namely Archaebacteria and Eubacteria. So they said all the living organisms they broadly classified into 2 super kingdoms, prokaryota and eukaryota. Prokaryotes they included 2 kingdoms. They are Archaebacteria and Eubacteria. And eukaryota they includes the protozoa, chromista, fungi, plantae and animalia. Now what is this chromista? Chromista they included all algae. So here this included the algae whose chloroplasts they contain chlorophyll A and C. So usually we have different type of photosynthetic pigments are there. Among them chlorophyll A is present in all the organism. So here those algae which had the chlorophyll A and C, they were given a separate kingdom and the name of that kingdom was given as chromista. As well as various colorless forms, they are closely related to them. So those organisms which included under this kingdom, they were diatoms, brown algae, cryptomonads and oomycetes were included under this classification. Next, we are going to study about red tides. What is red tides? Red tides is caused by a toxic bloom of dinoflagellates. Okay, so red tide is caused by a toxic bloom of dinoflagellates, that is a type of algae like Gymnodium breve and Gonialex tamarensis. So here this red tide is caused by a toxic bloom. Toxic bloom means the algae they produce a toxic substance. Because of that, what is happening? One major incident happened in the west coast of Florida in the year 1982. And as a result, it killed hundreds and thousands of fishes. So this was called, because of this toxic substance that gives a red color to the tide. As a result, it is called as a red tide, and this is because of the algae Gymnodium breve and Gonialex tamarensis.